Okay, well, once again, welcome to folks. I have about 2 minutes after the hour, so I think I'll go ahead and start with the introductory portion, which is going to be very brief, but that'll give a few more folks a chance to log in as we get started here. We're very fortunate to have an interesting subject today. We're going to hear about some ocean acidification information and research, something that's important to a lot of us these days. But before we get started, let me just say that the Pacific Ocean Education Team, or POET, is the group that sponsors these webinars. We're an interdisciplinary team of National Park Service staff from both the Pacific West and Alaska regions. And actually, we include a couple of other agencies too. NOAA has some folks that have become active members of the Pacific Ocean Education Team. The team was established in 2008 to enhance communication, education, and interpretive ocean stewardship efforts in the Pacific West and Alaska regions. We have a number of stated goals, including planning and scheduling ocean stewardship education sessions through trainings, workshops, webinars, and other distance learning presentations. And that's where our bi-monthly, or our monthly webinar series came from. We try to do it monthly. We can be a little hit and miss, but rest assured we'll be sending out emails when our next one is available, and we do have a couple things coming up in the next month. Past topics of this webinar series have included things like the benefits of marine protected areas, the vulnerability of coastlines to sea level rise, current population trends of endangered stellar sea lions, so we cover a wide variety of marine-related topics. Recordings of our past webinars can be found on the Ocean Alaska Science and Learning Center's YouTube channel. And if you just go to YouTube and search for Ocean Alaska Science and Learning Center, that's the organization I work for, and we house and host those recordings there. I'd also like to acknowledge Sarah Allen from the Pacific West Regional Office. She helps in coordinating this webinar series, and I'd like to acknowledge Yvonne Menard at Channel Islands National Park. She is the chairperson of the POET group, and she really is the driving force and energy behind this group. So we're very excited today to have with us Carol Blanchette and Lydia Kapsenberg. And Carol is a marine ecologist at UC Santa Barbara's Marine Science Institute. She studies the ecology of coastal ecosystems, including nearshore kelp, seagrass, and rocky intertidal habitats. And she is a co-PI on an NSF-funded research grant to study the impacts of ocean acidification along the U.S. West Coast. And with her today is Lydia Kapsenberg, who's a graduate student in Dr. Gretchen Hoffman's lab at the University of California, Santa Barbara. She's been studying coastal pH dynamics in Antarctica and in the Channel Islands National Park in California. And her interests are to understand how marine organisms are adapted to pH variability and to explore management practices that could potentially minimize the local effects of ocean acidification. So those are topics that are certainly of interest to a lot of us Park Service folks. So I'm going to turn it over to Carol and Lydia at this point and let them start the show.
Hi everyone, this is Lydia Kapsenberg speaking, and I'll be presenting the first part of the talk, and Dr. Carol Blanchette will talk to you after I do. And I just want to thank you for taking the time to listen to us talk about ocean acidification. So I'll introduce ocean acidification for those of you who haven't heard of it or only know some of the details about it, and then I'll talk about the variability in current ocean conditions, and I'll use my research from the Channel Islands National Park as an example. And then Dr. Blanchette will talk about the potential for evolutionary adaptation to this phenomenon and the role that marine protected areas can play in helping protect marine species for this in the future.
Lydia, can I interrupt you for just a moment?
Yes.
This is Jim Pfeifenberg, and I failed to say to folks, and most of you probably know this, you've been at webinars, but there is a chat box and a question box. You can use either one of those to pose questions to us. I'll be looking at both of them. Probably the question box is the best one to use. So if you have questions along the way, type them in there. We'll take some time at the end of the presentation to go through as many questions as we can get to. Thanks, Lydia.
So before I talk about ocean acidification, we really have to look at what's happening in the atmosphere first. And so here's a graph of CO2 in the atmosphere since the 1960s. And this plot of increasing CO2 is called the Keeling Curve, was started by Dr. Keeling, and it's atmospheric CO2 levels from Hawaii. And what you can see is that there's some annual variability that's pretty steady, but overall the CO2 has been increasing at a steady rate since really the Industrial Revolution. And this is due to human emissions of CO2 from fossil fuel burning and cement production. But the CO2 doesn't just stay in the atmosphere. So the CO2, as it's accumulating in the atmosphere, starts to dissolve into the seawater. And this is happening in the surface layer of the ocean, and it's very similar to carbonating soda. So it's the same process as forcing CO2 into the seawater. And this blue line here shows the CO2 concentration in the surface layer of the water in Hawaii. And what you can see that it's largely variable, but it's increasing at the same rate that it is in the atmosphere. The overall levels are slightly lower because there's a certain time delay for the CO2 in the atmosphere to dissolve into the water. And so the ocean is actually a really big CO2 sink. And 30 to 50% of atmospheric CO2 is absorbed by the ocean. And this reduces the greenhouse effect and the rate of climate change, which is really good, but this comes at a cost. And that cost is ocean acidification. And this is a decline in ocean pH. So now the green line here shows you pH data from Hawaii in the seawater. And this green data has a secondary y-axis called, it's listed as pH here, and it's this decline in pH, why we call it ocean acidification. So you can see that the water isn't actually acidic, meaning the pH isn't below this phenomenon. So ocean acidification is the increase in seawater acidity due to human-induced carbon emissions, and this is pretty well documented in various places around the world, and Hawaii is just one of those data sets. And so for those of you who are interested, I'm just going to go over the actual chemistry behind this to show you how it actually causes a decline in pH. So when CO2 enters the water, it mixes with water molecules, and through this reaction it actually creates carbonic acid, and this the carbonic acid mixes with carbonate ions and it produces these two bicarbonate ions. So we have increased levels of carbonic acid, we have increased levels of bicarbonate, and carbonate ions are being used up in the process. Well, carbonate ions are really important for other things in the ocean, mainly calcium carbonate. So any marine animal with hard body parts like oysters, mussels, sea stars, sea urchins, coral reefs, they all build their hard body parts out of calcium carbonate. And they use these carbonate ions to do that. But as the CO2 is dissolving into the water, these carbonate ions are being taken away to form other compounds. And so we have a reduction in carbonate ions, which is harmful for marine organisms because it's kind of a building block for them to build skeletons. But we also have more of this carbonic acid, which causes the pH to decline. And so the biological impact of this is that it's affecting marine organisms' ability to build calcium carbonate shells, or to build strong shells. And so I'm just going to give you two examples of this. The first one is for pteropods. So pteropods they're a free-swimming marine snail, and they create this very beautiful shell that's made out of calcium carbonate, and a lot of animals eat them. So they really sit at the base of the food web. Krill eat them, salmon eat them, whales eat them, and then this pictures below shows the shell of a pteropod exposed to see is that over time the conditions are corrosive to the shell. So even though that the water is not acidic, it can still facilitate this dissolution of the shell. And recently, well, in 2012, there was a paper published that has documented this shell dissolution on live pteropods in the Southern Ocean. So this is something that is really happening, and the pteropods are considered in the coal mine for ocean acidification, and they might be a real good indicator of the biological effects of ocean acidification. The other area that we've seen the effects of ocean acidification is in the shellfish industry. So there have been a lot of crashes recently with the shellfish industry in the Pacific Northwest where there have just been complete collapses of oyster seed production. And here are two pictures of a one-day-old oyster larvae, and you can see under the high pH photo that the shell is larger and it's very round and very even. Under the lower pH, which are natural conditions that have been occurring occasionally, the shell is smaller, it's a little bit irregular, and the shell is just not as strong as it is when the pH is high. So this has major impacts for food security, but also local economies and people who depend on these fisheries for their livelihood. And so, knowing that this is happening, in our lab we asked what's actually going on in the wild and what is actually the current pH. A lot of ocean acidification studies use stable laboratory pH conditions to try to replicate current and future conditions in the field, but these studies often lack information about the real ocean in coastal regions. So in my lab, we've deployed pH sensors in various places in the world. So here are 3 sites, one from California, temperate region, one from Hawaii, French Polynesia in the tropics, and some of our data from Antarctica in the Southern Ocean. And here the time scale is on the x-axis, and on the y-axis is pH. And you don't need to pick up too much detail except for the fact that they're all different. And so what we realize is that what's happening in the open ocean and what models are based off of to make these future predictions for ocean pH conditions may not apply evenly to all areas of the world. So there's this inherent variability among pH traces in different locations. So I was interested to investigate this further, and I have been deploying pH sensors in the Channel Islands National Park to look at the sources of pH variability that we can find in our own backyard here. And so here's a picture of the Santa Barbara Channel. The islands pictured here are the 4 northern islands of the Channel Islands National Park. And today I'm going to talk to you about the pH data collected at Anacapa Island and Santa Cruz Island. I've also started deploying oxygen sensors there, and they also measure salinity and temperature and pressure. And with more of this data, we can really try to identify the sources of this variability that we see in coastal environments. So here's a graph of pH data, and this is very typical for what we've been finding in the national park. So pH is on the y-axis again, and the x-axis shows time. This is about a 3-month deployment from last year. And the purple pH trace is from Santa Cruz Island, and the black pH trace is from Anacapa Island. And what you can notice right away is that they are very similar. They have this very strong diurnal cycle that is very persistent. And there's a little bit of other underlying variability going on. And so the main thing to note, though, is that these 2 sites still are a little bit different. There's the daily magnitude for Santa Cruz Island, for example, is a little bit larger than that of Anacapa Island. But overall, the pH is above 7.9. And just to give you some reference, the open ocean is considered 8.1 pH. And the predictions for the end of the century, assuming we produce carbon dioxide emissions at the same rate that we are now, the pH is predicted to be around 7.7. So that's below what I'm showing on this graph. And so what we think will happen is that this kind of variability will persist, but the overall mean of this pH will be much lower in the future. And so if we now look at the other data that I've collected, one of which is oxygen, we can plot that with the pH. And so this is just showing the pH data from Anacapa Island in black, and then on the secondary y-axis is percent oxygen saturation, and this is the blue line. And what you can see is that the blue line is nearly identical to the black line. So that means that dissolved oxygen is extremely highly correlated with pH. And because the changes in CO2 and pH that occur are independent of any sort of changes that occur chemically with oxygen, this makes us conclude that these daily cycles are driven by the same process. And that same process is photosynthesis, it's biology. So what we've captured in this variability is essentially the breathing of the kelp forests and these seagrass beds. And so here's just a zoomed in plot of that correlation. So pH is in red and oxygen saturation is in blue. And what happens is that during the day when photosynthesis is active, the kelp or the seagrasses are actively taking up carbon dioxide. So this is the reverse process of the chemistry that I showed you earlier. So this means that the pH is actually going up during the day. So carbon is taking up during the day, pH is going up, and for every carbon dioxide molecule that's taken up, one oxygen molecule is released into the seawater. And so that's what causes the oxygen to go up during the day also. At nighttime, when no photosynthesis is happening and the whole reef is essentially breathing, they're breathing out CO2. Accumulation of CO2 to do with ocean acidification in general, and it causes the oxygen to go down because the organisms are all using the oxygen in the water. So the pH variability in the Channel Islands National Park is quite largely driven by biological activity on a daily cycle. And the real question is, does this variability matter? So I had mentioned that a lot of ocean acidification studies use really stable conditions in the laboratory. So here is a study by Christina Frieder, who's a scientist from Scripps Institution of Oceanography at UC San Diego. And she conducted this really neat experiment to look at the growth of mussel larvae, so really similar to the oyster example I showed you before. It's a marine mollusk, and she grew larvae for 8 days under these 3 different conditions. There's condition of ambient stable pH conditions, which is closer to what the mean pH is in coastal regions in California. And she exposed larvae to future stable conditions of around 7.5 pH. And this third treatment where there was actually a semi-diurnal pH cycle, but the overall mean of the pH matched that of the future stable conditions. And then she measured growth. And she did this for 2 species, Mytilus californianus and Mytilus gallo-provincialis. And so the first, so this is a plot of shell length, so it matches the growth of the larvae. And so what you can see under ambient stable conditions in both species is that the growth is pretty high. But what happens when the larvae are grown under these low pH conditions of 7.5, the growth is reduced. And this is a phenomenon that we see among a lot of species, and it's why we conclude that ocean acidification makes it hard for marine organisms to build their shell. But the interesting thing is that when you expose these larvae to this variable future environment, you get different responses. So with Mytilus californianus, the larvae shells were still really small, but with the other species, with Mytilus gallo-provincialis, the larval shells were actually bigger than when they were just exposed to stable conditions. And so what this means is that variability can have an effect, and it's important for us to try to start looking at what this variability in ocean conditions means for future marine ecosystems. And so if we zoom back out from Southern California and we think about the variability that we've seen across the world, and even just within Southern California, there's a lot more variability happening than just these diurnal cycles. And so we're really interested to find out if variability in ocean conditions can help marine species adapt to future changes. So I'm going to turn it over to Dr. Carol Blanchette, who will tell you more about this.
Okay, thanks, Lydia. So this is the next part of what we'd like to focus on is exactly what Lydia was describing in terms of that variability. And there's two really important distinctions in terms of variability. There's spatial variability, and then there's temporal variability. And Lydia gave you some nice examples of both of those aspects of variability. As many of you are familiar, nearshore coastal ecosystems happen to be in an extremely variable environment due to a lot of the oceanographic processes that happen along our coast. These ecosystems also are incredibly important for a number of reasons. They contain a really high biodiversity of species. They have a high economic value for many fisheries that are important. A lot of the especially in California, the economically important fisheries tend to be in this very nearshore ecosystem. These are also ecosystems that are highly utilized for recreation. The vulnerable spot in these ecosystems is that they lie in very close proximity to where many human population centers are. So they're really susceptible to a number of impacts due to human pressures. So some of those things are pollution, are also intensified along the coast. And as Lydia mentioned, there's a lot of variability in ocean conditions due to temperature variability as well as the chemistry. And if we think about these coastal ecosystems in a forward-thinking sense, in terms of the climate change impacts that they're likely to experience in the future, they're clearly going to be hit with an array of probably fairly negative effects due to many things ranging from sea level rise to storm intensification. Many of you are probably familiar with the harmful algal blooms that can occur along the coast. And then finally, the one that we're really focused on is ocean acidification. So the real question, especially for people that really are tasked with managing these coastal ecosystems, is how will these organisms respond to something that we have very little control over? We can project and we can predict what the future ocean chemistry is going to look like in the future, and although we do have some control over that in terms of reducing emissions, even if we stop reducing emissions tomorrow, the CO2 is in the pipeline, so to speak, so the acidification of these regions is going to continue. And so we've really taken this approach of trying to understand how organisms are likely to respond to these conditions, even taking into account the fact that we can't really stop the changes that have already happened. So there are a variety of possibilities. Certainly organisms that have capability of movement could migrate. There's also the possibility that some organisms may just be able to tolerate these conditions. However, the third one is really sort of a very interesting idea, is that potentially some of these organisms may have the ability to adapt to this change. And there are a couple of ways in which organisms can adapt to change, and a lot of that depends upon the rate of change. Whoops. So I just want to take a step back before I start talking about adaptation, because I think it's really important to consider how evolution works in order to understand adaptation. So we know that organisms can adapt to environmental change, and we've seen this happen throughout the history of the planet. We know that that adaptation requires really two things. It requires sufficient standing genetic variation. So we need to have a lot of genetic variability in a population. We also need to have the potential for a high intrinsic rate of population growth for adaptation to happen. Phenotypic plasticity is one way that populations can be buffered against these immediate impacts, and phenotypic plasticity also allows for genetic adaptations to catch up. So it gives organisms a way to deal with change outside of adaptation. We also know that selection through the process of evolutionary natural selection occurs when there is that phenotypic variation among individuals, and some of those phenotypes have higher fitness than others. So we know also that traits such as those required to cope with climate change will not evolve unless there is that sufficient heritable genetic variation and in the direction in which selection acts on those traits. And so this is really pretty fundamental evolutionary biology, but it's really the aim of global change biology is to understand how this happens. So one way to think about that is to estimate the level of genetic variation that exists in populations today in traits that are essential to coping with climate change. So if you look at a population, you might ask what sorts of genes are in the standing stock of the population that might confer some ability for that organism to be able to survive a certain amount of, let's say, temperature variation or pH variation. We also want to look for places where we expect to find a great deal of variability in those traits that might be related to climate change. And so sort of what this is all leading up to is the idea that if we're going to look for a place where we think organisms might have the greatest potential for adaptation to climate change, the best place to look is an environmentally variable ecosystem. And so a lot of what Lydia just described about the spatial and the temporal patterns of variability in the Channel Islands is something that's really characteristic of the U.S. West Coast. And much of the U.S. West Coast is what we refer to as an upwelling ecosystem, so that along it's an eastern boundary ecosystem, which means that the waters from the Pacific Ocean, due to the rotation of the Earth, are brought up along the coast, moved offshore at the surface, and so those waters that are drawn up from depth tend to be really undersaturated. So those are waters that are naturally more acidic than most of the ocean. They're low temperature, and they also happen to be low pH. And that those waters, this figure just shows you a relative model of upwelling along the coast of California. The reddish areas are areas of strong upwelling, and you can see that those areas are places where the pH is quite low relative to the surrounding water. And so this process of upwelling, again, is just driven by the rotation of the Earth. It's driven by wind patterns, and that upwelled water that comes very close to the shore tends to be what we call cold and old, which means that it's really low temperature because it's brought up from depth. It's old, meaning that it's been the residence time of that water, it's been in the ocean for a really long time. And so that's why it tends to be high in nutrients and dissolved CO2. It has low oxygen and low pH. And so this is also related to the idea I just was talking about. Even if we stopped our emissions tomorrow, there's a giant pool of that water that's already dissolved CO2 for a really long time that's still in the system. So no matter what, we are going to continue to experience ocean acidification into the future. So your attention to one part of it, that the only main point I want to make is that it's been recently pretty widely recognized that upwelling ecosystems like the California current are actually some of the most susceptible ecosystems on the planet to the continuing and future effects of ocean acidification. So the x-axis in this figure shows you an increasing scale of atmospheric carbon dioxide, and you can see that the y-axis represents the saturation state of aragonite, and the other side represents the saturation state of calcite. When that saturation state goes below a certain value, then the water, as we call it, becomes undersaturated. And you can see that there are a couple different ecosystems represented here. There's the tropical ocean, the Southern Ocean, the Arctic Ocean. California current system is in red, and it is, as we go through time, presumably, as we add more CO2 into the system and go along the x-axis, the California current is really pushing close down in well below the saturation horizons for aragonite and calcite, meaning that it's probably one of the places in the world that's going to be likely to be impacted first due to ocean acidification. So how does this really relate to the question we're trying to get at, which is how can organisms potentially adapt to this variation? So this is one of the research projects that I'm involved in, and one of the key questions of that project has been, is there local adaptation in key marine invertebrates as a result of this variability, this mosaic pH substitute this spatial variation for temporal variation, in the sense that we're trying to use the variation that exists along the California coast to ask questions about what might happen in the future. And this panel just shows you a range of our study sites across Oregon and California. And just to mention also, the two organisms, the invertebrates that we've focused on for a number of reasons, are mussels and urchins. And Lydia talked a little bit about mussels, and I'm actually just going to present some data on the urchins. So how do we measure this gradient of ocean acidification? We actually have used sensors that have been developed through a collaboration with scientists at MBARI, which is in Monterey Bay, who have built basically pH sensing instruments that we can mount on moorings offshore, and we can also mount them in tide pools in the intertidal. Because what we're really interested in is looking at that water that's really, really close to shore, and as it moves from sort of offshore to onshore. And this is a lot of data, and it's a bit of a busy figure, and all I want to do is show you the results of the patterns that we've been seeing across this mosaic of variability. And so we have basically mooring traces and intertidal traces from each of these sites through time. And if you remember from the data that Lydia presented, you know, some of the pH values that she was looking at fell around 7.9 at the Channel Islands. And if we go across the west coast here, we see a really broad spectrum of variation in pH. And it does tend to be correlated, as we sort of were thinking about, with the wind patterns, which are really what drives the upwelling. So we've taken all of the data that we've collected from these sites along the west coast, and we've tried to calculate the frequency of time that each of the sites would spend at a pH that's at a value of 7.8 or less. And the reason we've chosen 7.8 is just because that seems to be a pH value where many organisms have started to really show deleterious effects. And so we can rank these sites in terms of that frequency of time that they spend at those pH values. And so one of the postdocs in Gretchen Hoffman's lab did a study looking at using genomics to assay the different kinds of genes that were related to this gradient in pH exposure and what kinds of activities those genes performed. And what he found was that there's a pattern of gene expression that was really highly correlated with that gradient of pH conditions, and that many of those genes were genes that did things like altered the bioavailability of calcium and adjusted the skeleton pathways to sustain calcification at low pH. So it was really suggestive that the larvae of these urchins were using really different strategies for dealing with this pH stress. The next example I'm going to show you comes from another postdoc at Stanford who is also part of our research group. And what they've actually seen is, again, looking at urchins, these are larval urchin forms, and they've done a number of studies where they've collected urchins from those sites that are spread out along the west coast that experience these really different pH patterns, and they've raised those larvae, and then they've also exposed larvae from these different families that have been raised from different sites to different levels of pH in laboratory conditions. And the main point I want to show you to focus in on here, on the left panel where you see the bar chart, the vertical bar chart, there are basically two different exposures of CO2 conditions that would be representative of a really high and a low pH condition. And the main point there is just to say that the larvae, in terms of their morphology, these are lots of different measurements of morphology, showed very little difference between those treatments. However, when they looked at the pattern of the genetic changes in response to those conditions, there were really, really large changes in the frequency of different alleles that tended to really important to an organism that would be calcifying. And so the result of this study was really that even though these urchins didn't really show a very strong response in terms of their morphology to those pH conditions, their genetics showed a huge change in response to those conditions. And so that urchins, which have evolved in this highly spatially variable ocean climate, really have this great capacity to evolve in response to those to future OA conditions. Oops. So kind of winding up here and getting down to what this really means in terms of management, what we like to think about is, you know, this question: how can we potentially manage for ocean acidification, given that organisms have this great potential to evolve, especially those that have been sort of experiencing this wide range of variability throughout evolutionary time. Well, one thing that's really important is the rate of change. And so the rate at which the environment changes is going to strongly affect the capacity of those populations to persist for long enough times for evolution to catch up with climate change. And so in terms of management, there's really two things that we think are important. Obviously, the first one is one you've all heard about: global actions to slow the rate of climate change. So, you know, reducing CO2 emissions is the only way that we're actually going to reverse what's happening in the ocean. In the meantime, though, one strategy that might prove to be really beneficial, given what we know about adaptation, is to conserve that evolutionary potential in organisms. And we know that marine protected areas have been established along the coasts of many states. California has been really leading this effort in recent times, and we now have one of the largest networks of marine protected areas in the world. And marine protected areas are really one way to conserve that evolutionary potential. And the reason why is basically what I just described. The only way evolution can catch up with climate change is if there's large enough population sizes. And MPAs are really implicitly They can provide some resilience in the face of climate change, and that's just one example of why they can do that. Another example that we haven't really talked about much today, but is also just in terms of producing or protecting really important habitats. So species like seagrasses and kelp that are really important in terms of biological regulation of ocean conditions are important species in these marine protected areas. And just as to tie this back to what Lydia was talking about, this is a map of the Northern Channel Islands, and actually many of the Northern Channel Islands contain large areas that are in these marine protected areas. And so this is just a great example of how management for one problem, which is overfishing, can really be a win-win situation in terms of helping out species in terms of their ability to cope with climate change. So we're going to sort of end our presentation here, and we certainly would welcome questions. We would certainly like to acknowledge many of our funders, including National Science Foundation, the George Melendez Wright Climate Change Fellowship, Seabird Electronics, Southern California Research and Learning Center, and the UC Ocean Acidification Center. It takes a lot of people to do this sort of work, and so we have many great collaborators and many great agencies that we've worked with. And what we'd like to do is just leave this slide up for you guys all to read and look at and think about. And these are really just, I think, what we feel like are the main conclusions of a lot of what we've learned over the last couple of years in terms of ways that MPAs and other conservation measures could be beneficial for providing strategies to allow organisms to adapt to climate change, and just different ways to think about that variability being
Okay, wow, very good. Thanks, Carol and Lydia, and I do apologize to everyone for the occasional audio glitching. I think a couple people commented on it. We got we got 90% of what you guys said. Every now and then there was a little hiss and audio glitch, but I'm thankful that in many cases I think the point you were making was reflected in some text on the slide. So for the most part, I don't think we lost out on much. And fascinating presentation. If folks do have questions, please put them into the question box, and we'll go through those. In some ways, this relates very directly to an article I just saw the other day about small ocean algae that apparently are reflecting a much higher ability to adapt than had been detected before to some of these projected changes. So really interesting stuff, and in some ways really sort of reemphasizes to those of us in this line of work the importance of maintaining those population sizes and maintaining those areas that are conserved and set aside. So let me get to some of the questions that we do have in the box here. One is asking about a term or phrase that you used. What is phenotypic plasticity? Can you expand on that?
Yeah, so phenotypic plasticity, I guess we could maybe providing an example would just be useful here. You know, one example could be the color or shape or size, or sort of like the outward form of an organism in terms of what it looks like. So how variable is it in any particular element of its morphology, its body form, its color, its shape, its size, and how different levels of variation in that phenotype can be selected upon either positively or negatively through evolution.
Okay, thank you. I hope that answers the question for for the asker there. And then another question, there are a couple folks who had this idea, by how old, by saying that the upwelled waters are old and cold, how old do you mean? In other words, could those waters reflect some sort of a pre-OA, condition? Are they that old, or how old?
Well, this is Lydia. This is something that's sort of hard to really track. It's difficult to track, say, one water molecule, but I've heard ideas that it's about 50 years old. And if you remember in the very beginning of the presentation, I showed you that the CO2 concentration in the water is increasing at the same rate as the atmosphere, but the absolute concentration is lower because there's this delay. And so there's a delay in the signature of CO2 in that deep water, and there's a delay in the time that that body of water was exposed to the atmosphere. And as it's moving around the global ocean, it's also accumulating a lot of CO2 from all the organisms that are respiring, and deeper water is known to be more acidic. So it would not, in the sense of, like, can this older water not have an ocean acidification signature? It's not like we could get deep water of high pH, for example. So what we see is that it's always low pH water that's coming up to the surface, and it's older, but by multiple decades, but not in a way that would really help in terms of ocean acidification.
And that's, and I think that's also reflective of why the idea is if we stopped producing carbon-based emissions tomorrow, and we reduced all of that CO2 that we're putting into the atmosphere, ocean acidification would continue through time, and the waters that are being brought up from depth would continue to be more and more acidic through time until we sort of ran out that cycle of water that's already in the ocean.
Right. Excellent answer to that. I appreciate that. Here's another question. What's the correlation between ocean temperature and acidification? In other words, is acidification going to be felt more strongly in colder or warmer areas?
So this is getting really into the chemistry, which I think is fun. Dr. Blanchett mentioned aragonite saturation state, and so this is a measure that relates directly to that dissolution of calcium carbonate that I talked about, and it's a phrase that's used a lot with ocean acidification, but it's not directly a measure of pH. Well, temperature effects on this saturation state, so this threshold of when we think things are going to be really bad for organisms, is also very temperature dependent. And so with colder water, this saturation state is lower. And so if you remember Dr. Blanchett's graph of sort of predicted ocean acidification trends in different ecosystems, tropical regions were actually really high. And polar regions and upwelling systems were one of the first to really cross this threshold of aragonite saturation state. So this is why polar regions are often considered to be the areas in the world where the effects will be observed first. And this is, I think this is why we've seen some of the first evidence of ocean acidification in the wild on pteropods in the Southern Ocean. And also we're beginning to see them in the upwelling ecosystems by the same group of researchers off the California coast. So that's kind of how temperature plays into the direct chemistry and how it relates to the calcium carbonate of marine shells. The other thing is that cold water can hold more CO2, so it's also the Southern Ocean, for example, is a little bit lower in pH than the open oceans just because it's very cold, and so it has a capacity to hold more CO2.
So that corresponds with explanations that I have heard before, but the slide that you just pulled up again, correct me if I'm reading this wrong, to me I see this showing the California current area as more susceptible to being undersaturated than the southern or Arctic Ocean. So that goes a little bit against what I just think I heard about colder waters. Or is the California current colder than the southern and Arctic Ocean?
So it's not colder, but it's this upwelling phenomenon that brings this extra lower, which is why in this, so that top red line within the Central California current system is the depth of, it's the surface layer of the ocean from 0 to 60 meters.
Mm-hmm.
And as you go deeper, the temperature is colder and there's more CO2 in the water, so the pH is lower. And that's why those bottom two red lines are below the surface layer line from 0 to 60. So these are all projections still, and so the important part of this message is that not all ecosystems are created equal, and different effects will be found in different ecosystems, like if you're looking at the same moment in time. And the Southern Ocean and the Arctic Ocean and upwelling regions are just one of the most vulnerable ecosystems for ocean acidification.
Okay, and I see, as I look at this again, I realize what you're showing is that the Southern Ocean, Arctic Ocean, and the Central California current system track very closely, actually.
Yeah, compared to the tropical ocean.
Gotcha. Okay, that makes more sense to me now.
There's also one additional aspect in the California current system due to that high upwelling, is that there's an extremely high amount of productivity in the California current system, and so with a lot of that biological productivity also, as things like this phytoplankton flues sink to the ocean, there's a lot of biologically driven reasons for driving that saturation state down as well.
Okay, we've got time for a few more, a couple more questions, and we do have a few more. What are you finding in coastal glaciated environments? What are you finding in Antarctica, for example, for ocean acidification results?
So we have been working in the Antarctic since 2010, and we have been doing two things there. We've been growing sea urchin larval cultures under what we think are future conditions for that region, and we've also been deploying pH sensors. And the data that I showed on that global graph, let me pull it up. So this is the middle panel. This is a really short deployment. So here we can see that the pH is extremely stable. It's about 8.0, and this matches what we see for more open ocean areas. But this is only during the month of November, which is the spring season in the Antarctic. And at this point, the ocean where we are is actually covered by sea ice. And we're just coming out of winter conditions at this point that are very cold. The sea ice is very solid, so there's not too much interaction between the atmosphere and that surface layer of water in terms of gas exchange. And I didn't show you the data, but we've left a pH sensor out there for 7 months, so that captures this summer season. So in Antarctica, we have this period of 24-hour daylight. So it's very different from, say, the Channel Islands, where, you know, the day is on a 24-hour cycle with 12 hours light and 12 hours dark. And so what happens in the Antarctic is that as soon as we hit this period of 24 hours of daylight and a little bit of, the ocean starts to warm a little bit, and it sort of starts this really, really extremely productive season. So huge phytoplankton blooms start to happen in December, and they peak in January. So this is data that is not here, but what happens is that the pH goes up. So it goes up to about 8.3 or 8.4, which is actually off this chart and higher than what we've observed at these other regions that are shown on this figure. And then after January, the pH actually starts declining, but the variability during the summer season is the same kind of variability we've seen in California, for example. So what the Southern Ocean is really considered this stable ecosystem, but what we found is that there's this really big annual cycle in pH. And you can think of this as one really long day. So in this one really long day that is the summer season in the Antarctic, we get all this photosynthesis happening in one day in California. And then into the winter, as soon as the sun starts setting and we hit this period of 24-hour darkness, respiration is the only biological process that's going on. And so that's putting a lot of CO2 back into the water, so that gets the pH back down to this 8.0 baseline. So that's what's going on in the water. What we found with the sea urchin larvae is that when we grow them under lower pH conditions like 7.7 or 7.6, they're a little bit smaller than when we grow them at 8.0, which is the conditions that they're actually exposed to because they develop during this spring period, this November period where we've shown pH for.
Okay, thank you. We have two more questions in the box. I'm going to try to squeeze them both in. It's coming up on the top of the hour. In terms of impacts to marine invertebrates due to climate change, do you see thermal stress as being more, less, or equal in impact to low pH?
You know, that's a really good question, and it totally varies on depending what species we've looked at. The range of variation in responses of marine organisms to stresses is huge. I mean, as you know, there's a lot of marine organisms that actually respond really positively to decreased pH. Not a lot, but some. And so one of the big issues right now people are really taking into account is this idea of multiple stressors. And so some organisms, you know, temperature's a much bigger problem than pH, and some of them it's really the interaction between temperature and pH where we see effects. And so I think we're sort of in this realm of ocean acidification research where people are moving beyond these experiments looking at different ranges of pH and trying to incorporate these multiple stressors to look at these interactions. And so, you know, certainly in places like the tropics, where although there's a huge amount of corals, which are incredibly important reef building invertebrates that have calcium carbonate hard parts that would be susceptible to pH, you know, right now they're actually facing a lot of stress due to temperature, and that could change through time as ocean acidification really starts impacting them even more.
Okay, I really appreciate you guys' time. I think we're not going to take time. There's only a couple more questions hanging there. They're good ones, but I feel like we need to let folks go. And really appreciate your time. Again, this session has been recorded. Thanks to the Poet Group for helping put together this webinar series, and a huge thanks to Dr. Blanchette and Lydia Kapsenberg for sharing their information with us today. Keep your eyes on the email. We'll send out another notice when we have another webinar coming up.
Great, thanks.
Thank you.